webinar, Cape Unit 2 Biology, and congratulations on your Cape Unit 1 Biology passes, and you're in your final lap now for high school. And for the persons who are in the seminar right now, I know it's because you want to secure a grade 1 for Unit 2 Biology, and more than likely to matriculate in your program of choice, and you want to earn a grade 1 because I'm sure you're confident that you can pass right now, having gone through Unit 1 Unit 1 Biology. So I don't think you're worried about passing the subject. I think what you want is to maintain. If you got a distinction in Unit 1, you want to maintain that for Unit 2. Or if you didn't get it for Unit 1, you definitely want to get it for Unit 2, because getting a distinction in the CAPE subjects, it makes it easier for you going forward. If you're seeking for funding or a scholarship, or if you're applying to a very competitive program, that grade 1 is going to help you a lot, going to save you money and time. So what ends up happening though, guys, is when persons get a grade 3 or 4 or 5, it is considered as a pass, right? It is definitely considered as a pass. However, the challenge is that usually you're not seen to be as competitive. So for that reason, it's great that you are here. And I want to just let you know what to expect in a Unit 2 biology syllabus. So biology is the scientific study of living organism, and the study of these subjects leads us to an understanding and appreciation of the concepts of life at all levels, and hence to a greater respect and reverence for life. And definitely Unit 2 biology is all about that, teaching you how to respect and have a greater reverence for life. The interconnected web of life and the unique role of the human species are integral to the dynamic value of the biosphere. The CAPE biology syllabus prepares students to acquire knowledge about how to protect, sustain, conserve, and improve the variety of life in the ecosphere. So why do we study biology, guys? Let me show you this clip.
We've got 60 seconds. Are you paying attention? Biology is the reason your body works and looks the way it does. The reason that your heart beats and your lungs breathe without you even thinking about it is because of your brain and nervous system. The reason your eyes and hair are the colour they are is because of your genetics. The reason you can eat an apple and turn that apple into enough energy to go for a walk is because of all the body systems, like your gut and your blood system, that can turn that matter into energy and that energy into movement. The reason that you age is partly biological. Whether you become 100 or not is down to your biology. The reason you walk, talk, are happy or sad, the reason you are you is because of biology.
So you will see that basically everything about our physical structure and behavior as human beings is determined by our biology. So in Unit 2 biology, we have 3 modules, 1, 2, and 3. Module 1 is focused on bioenergetics and conservation. Module 2 on biosystems maintenance, and Module 3 on the applications of biology. So what are the things we'll cover in Module 1? We will learn first about photosynthesis and the production of ATP. And you notice that there are not many topics in the module. What it therefore means is that we're going to be going in great detail. We are going to go into granular detail because we have less topics. So for photosynthesis, we're going to learn about the structure of the chloroplast. We're going to learn exactly the mechanism by which we capture solar energy to excite electrons from the chlorophyll molecule, and how we pass those electrons along electron transport carriers to generate electricity to make ATP. So we're going to learn the process of photosynthesis in granular detail, from the light-dependent to the light-independent stage. We're going to learn about the limiting factors of photosynthesis, how greenhouses work. So that will take us some time because we're going into such great detail. Then we're going to learn about cellular respiration and how to make ATP. So we're going to learn about the structure of the mitochondria. We're going to learn about the 3 stages of respiration: glycolysis, Krebs cycle, electron transport chain, learning about anaerobic versus aerobic respiration, and in any way that we can apply the knowledge, we will apply it. Then we learn about how energy flows through ecosystems, through the food webs and food chains, And we're going to also learn how to cycle nutrients in our ecosystem. And then we're going to study the structure of ecological systems, how we get biodiversity, how we have different species living in an ecosystem, how we have different habitats in an ecosystem, and more importantly, how do we conserve biodiversity and conserve our ecosystems? Because we're going to learn the value that our ecosystems have to us. So module 1 is a very interesting module, going through these 2 cellular processes. Then we move on to ecology, learning how we impact our environment. So that's module 1. So I have this video for you to look at for photosynthesis. So this experiment was basically showing how we had different rates of photosynthesis by varying the percentage of carbon dioxide in the atmosphere. And they showed that by increasing the carbon dioxide level by 3 times in the atmosphere, there was a significant difference in the growth or germination of the seedlings. So it's definitely showing you that high carbon dioxide concentrations do cause an increased rate in photosynthesis. But at the KP level, we have to be able to explain how and why. We have to talk about exactly what phase of photosynthesis are we increasing with the increased carbon dioxide levels. So we're going to go in great detail for photosynthesis. And then next, we're going to look at cellular respiration. And this is a video showing you how our knowledge of cellular respiration can be actually applied in real life.
Hey everyone, we're here with another video. We have all watched that movie scene. And you would be wondering, how did he die so fast? Bullets take longer to kill. Well, to understand that, you need to know the mechanism of cyanide poisoning. It's much simpler than you think. Human body is made up of cells. Except red blood cells, all cells have a part called mitochondria. Mitochondria is responsible for production of energy. Now, imagine a cell to be a car. The mitochondria will be its engine. Like an engine, it needs air or oxygen. But instead of petrol or diesel, its fuel is glucose. When cyanide enters into body, it inhibits the supply of oxygen to the cell by blocking its path, which chokes the cell and stops the energy production. As the cell is devoid of energy, it dies. The same thing occurs throughout the body really fast, killing the cells, and ultimately the person dies. Though the exact time of cyanide poisoning depends on a lot of factors like dosage and body weight. Different sources say it can take from several seconds to several minutes. Cyanide.
So guys, you see how it is that respiration is so critical for us, right? Because inhibiting one enzyme in the respiratory chain of respiration causes death within seconds, because the enzyme that is being inhibited by the cyanide is an extremely important enzyme in the production of ATP. And without the production of ATP, and our cells can't function because every single cell requires energy, you find death within seconds. So this is one of the ways our knowledge of respiration is applicable, whether in a good or bad way, because cyanide can be used as a poison to kill people or taken voluntarily as seen here. But of course, we're going to learn about the stages of respiration, the critical enzymes involved. And so by the time we are finished, we can understand the mechanisms of how we can make it more efficient or less efficient. And then after we're finished with photosynthesis and respiration, then we look at ecological conservation. That's when now we are more concerned with understanding the environment around us. And in the unit 2, module 1, we are going to understand that as human beings, we are not the only important species on this planet. And I think this is recently that we have really come to acknowledge it, that every single living organism on this planet has a purpose and has a role. But before, when we could not find a direct purpose or a direct role for something, we thought it was useless and we thought it was dispensable. But it was only after certain species that we considered to be useless started to become extinct that we realized that even when they don't have a direct benefit to us, they have a role in the ecosystem in sustaining the stability of the ecosystem, and we benefit from the stability of the ecosystem. So whether we like it or not, indirectly, we are dependent on every other species on this planet. So therefore, it is in our interest to preserve them and to allow them to survive. They have the right to life as much as we do. And that is a principle of ecological systems, biodiversity, and conservation that we're going to be exploring in module 1. So let me show you this clip. All right, guys. So you would have seen the reasons why, the fact that when other species die, it should concern us. So if there is a species of grass somewhere in Alaska that is becoming extinct, we are understanding that that is our problem. Even though we could see no possible reason why the species of grass in Alaska should be of any significance to us. What we are understanding now is that that species had a role in its ecosystem, and that ecosystem had a role on this planet. And it will eventually affect us, whether in the immediate future or the distant future, right? So we're going to learn that. So then after we finish with module 1, we go to module 2. Module 2 now in unit 2 biology deals with biosystem maintenance. So we're going to be learning about how we uptake and transport water and minerals in plants. And as we said, detailed study to the granular level. So we're going to learn the details of the structure of the xylem vessel, how we pump ions into the roots of the plants, exactly by what mechanism water is able to move from the roots all the way up to the leaves. You would have been introduced to it in CXC biology, but you're going into much greater detail now. We're also going to learn about how we transport sugars and amino acids in the phloem vessels by another process known as translocation. So these two transporting plants will take up the first 2 topics in module 2. Then we go to the circulatory system of mammals. This is quite interesting because we're going to learn so much more about the human heart, about electrical conductive system in the human heart, the structure of our valves, learn more about the structure of our blood vessels. And then there's something else we're going to be introduced to in CAPE that we never got in CXC, We're going to learn about the more detail about the molecule of hemoglobin and how it is this special molecule designed to transport oxygen. Unlike any other molecule we have in the body, we're going to learn about the oxygen dissociation curve and hemoglobin. And after that, we will learn about homeostasis and hormonal action. We will learn about how do we maintain a constant internal environment. We understand now that we have to maintain levels, the temperatures and calcium levels and levels of ions, etc. They have to be maintained at a steady level in the blood to allow our enzymes to work optimally and allow our systems to work optimally. So we're going to learn the mechanism by which we maintain homeostasis and learn about hormonal action. Then we have to learn about the kidney, excretion, and osmoregulation. Very interesting one topic this is as well. We're going to learn again granular details about the process of excretion, urine production. How do we regulate how much urine we produce on a hot day or on a cold day? How the hypothalamus works with the kidney. So you would have been introduced in CXC, but going in more detail. And finally, in module 2, we learn about the nervous system, nervous coordination. Learn again about the neurons. We're going to learn in granular detail how our neurons are able to generate an electrical impulse. We're going to learn that it's not actually, well, at the higher level, we don't call it an electrical impulse. We call it an action potential. And you're going to have to learn exactly how an action potential is developed and how it is propagated along a nerve or neuron. And the synapse, we're going to learn what happens in the gap between two neurons and how that can be manipulated to make weapons and drugs and medications as well. So this one, topic 6 in module 2 has a lot of applications. So let's learn a bit of some of the applications of some of these topics in module 2, starting off with the circulatory system in mammals.
Orthotopic heart transplantation. Heart transplantation is the process of removing a diseased or failing heart and replacing it with a healthy heart from a donor. In order to remove the recipient's heart, aortic as well as superior and inferior vena cava cannulation is performed so that cardiopulmonary bypass can be initiated. Once on bypass, a cross clamp is placed across the aorta and cardioplegia is inserted through the aortic root so that the heart is stopped for removal. Caval snares are placed around the superior and inferior vena cava cannula. The SVC and IVC are divided, leaving a generous rim of tissue to which the donor SVC and IVC will be sewn. Next, the ascending aorta and pulmonary arteries are divided. The left atrium is divided in such a way to leave the 4 pulmonary veins attached to a cuff of left atrium, where the donor left atrium can be attached. The failing heart can now be removed from the patient. The healthy donor heart is then brought into the field and examined for any heart defects. The left atrium is sutured first. Next, the ascending aorta is sutured. Once the aortic cross clamp is removed, normal cardiac activity resumes. The pulmonary artery, inferior vena cava, and superior vena cava are sutured to the recipient's corresponding structures in this order. Once completed, we wean the heart from cardiopulmonary bypass and ensure by echo that all connections are open, that all valves are functioning well, and that the function of both ventricles is good.
All right, guys. So I was showing you this because it was an application of the knowledge of the human heart. So because we are so versed in the structure of the human heart that it allows us to do these things like transplants. And he was even showing that I don't think this was a human heart, but it was basically showing you the great vessels of the heart, which I want you to see. That was the main thing. All right. So Next thing we're going to, as I said, one of the topics we learn in module 2 would be the kidney excretion and osmoregulation. This is showing you the negative feedback pathway for how we control the amount of water in our urine. If the water levels are high, then we don't produce the hormone ADH and we produce high volumes of urine. But if the water level is low or we have too much salt in the blood and the water potential falls, then the brain releases ADH, which causes more water to be absorbed in the kidney and we get less concentrated urine. But this is a simplified version of it. We're going to go in greater detail. Nervous system, as I said to you, this one has a lot of application. I wanted to share this application with you about the use of a nerve agent. And this is because we study the nervous system so well. And we understand that there's a gap between our neurons, which is the synapse, and that a lot of chemical agents and weapons can target the synapse.
Getting elected president.
So you see how these nerve agents were used by Saddam Hussein against the Kurdish people. All right. And this was all based on knowledge of the nervous system. So finally, the last module of Unit 2 biology would be based on the applications of biology. So in this module, we have 4 topics: health and disease. We first understand what is health and disease, which would obviously be the absence of health, or there is suboptimal health. So we're going to learn about the different categories of diseases and what are the determinants of health. We'll understand that health is not just based upon your genetics. Health is based upon your environment, your level of education, your availability or your access to clean water, food, education, empowerment, politics. So we learn that health has to be considered on a much broader scale than just, being sick in the hospital. So we understand what are determinants that can affect the health of a population. Then we're going to learn about immunology, which I think most students find this topic to be most exciting in Module 2, when you learn the details of how our white cells work to protect us against pathogens. We learn about the innate immune system, which is our white blood cells or phagocytes, which just try to engulf pathogens as soon as they breach our first line of defense. And then we learn about our lymphocytes that are way more specific, that produce, B lymphocytes produce antibodies, and T lymphocytes are able to directly attach to cells and kill them. So we're going to learn that very elaborate, intricate system that we have in immunology for defending us against pathogen, and even how we can use our knowledge of immunology to make monoclonal antibodies. And monoclonal antibodies are very useful because they help us to diagnose pregnancy, diagnose HIV. It helps to find cancers in the body and treat cancers. So that is going to be a very exciting topic in Module 3. And then we go to social and preventative medicine, where we understand the factors contributing to several diseases. So we're going to study diabetes, we're going to study hypertension, we're going to study HIV/AIDS, we're going to study dengue fever, and we're going to study atherosclerosis, obesity. We're going to understand what are some of the social factors which lead to these diseases, what are the complications they cause, and what can we do to help to prevent the prevalence of these conditions. So that's social and preventative medicine. And then our final topic is substance abuse. We learn about alcohol abuse, smoking, Under this topic, we also learn about the physiology of exercise, a little bit about that. So that's module 3. So I want to show you this video for immunology, because immunology became so important during the COVID-19 vaccine. If there was no other time it was important, it was during this pandemic that the knowledge of immunology saved us. All right. So I want you to look at this clip.
The U.S. authorization of 2 COVID-19 vaccines marked a turning point in the pandemic and vaccine science. These vaccines use messenger RNA, or mRNA, to deliver immunity. mRNA are short strands of instructions used to build proteins. It's impossible for these mRNA vaccines to alter our DNA in any way. Although that technology has been in development for decades, mRNA vaccines hadn't made it to market, until now. There are a few ways to make a vaccine. Most use a version of the pathogen they are trying to protect you against, like the flu or measles, to trigger an immune response. You can think of vaccines like a biological wanted poster. They show our bodies how to recognize the enemy, an antigen such as a harmful virus or bacteria, so we know how to react if it ever shows up. Our bodies will be prepared to send T cells to attack infected cells, and B cells to produce antibodies, which neutralize the virus and prevent it from invading healthy cells. mRNA vaccines use innovative technology to deliver protection. If traditional vaccines deliver wanted posters, these vaccines carry USB drives with instructions that tell our cellular machinery how to print its own poster. In other words, they instruct the body to make its own antigens. In the case of the coronavirus, the antigen is the infamous spike protein that attaches to and infects your cells. These vaccines use mRNA, a molecule that our cells need to create any type of protein to introduce us to that spike. To start, researchers isolated the part of the coronavirus's genetic sequence that codes for the spike protein. Then, they put an mRNA molecule containing the code into a super tiny drug delivery system known as a lipid nanoparticle, which can enter our cells. Inside the cell, ribosomes use the code to create a protein chain that resembles the enemy, the coronavirus's spike protein. Those cells can then temporarily display a lookalike of that protein, which prompts our immune cells to respond as if it were an antigen from a traditional vaccine.
All right, so I want to also show you this clip on underneath social and preventative medicine, which is basically, as we said, we're going to be studying conditions such as HIV, dengue, obesity, hypertension, to understand what are the factors which result in these diseases, how they affect the population, and what are ways we can do to prevent it. So this is a video I wanted to show you on HIV.
This was the moment when a little boy shamed the world. Nkosi Johnson's dignified demand for AIDS drugs for all and an end to stigma.
We are human beings. We can walk. We can talk.
Nkosi died shortly after, but not in vain. South Africa is turning a corner. 16 years on and millions of lives later, the rules of the game have changed in South Africa. Young men like Sanelli do now have HIV drugs, enabling them to do the things they love.
I was unfortunately born with it. Got it from my mother. I grew up in a family of 3 sisters, 1 brother and me and my mother. Never knew my father. I lost all of them, and the last one was doing what you're doing, journalism. She was fighting for our success. Unfortunately, she also died. So now it's between me and my nephews to build each other and become a stronger family. Yeah.
With the support of friends, Sanelli's had the courage to speak out. Infected as a baby back in the day when leaders here were still in denial about the virus which causes AIDS.
I never asked for any of this to happen in my life, to lose my parents, to lose my financial helper, stuff like that, you see. I never asked for them to happen in life. So it happens, it happens. I have to live with it and be strong.
And he knows a new generation can't afford to take their eye off the ball. It's a mixed picture. Fewer babies are now born with HIV, but 2,500 young South Africans are becoming newly infected every week. This home where Sanele grew up was set up by Nkozi's foster mother, Gail. Virtually all the youngsters are HIV positive.
Good girl.
The good news is that they have the drugs to keep them alive. The bad news is that stigma and shame still persist, pushing many into silence.
They're still very nervous and very embarrassed about coming forward because they will be judged, they will be ridiculed, and they will be maybe rejected.
Yes, I had sex with Brave. Did you use a condom? She says no.
Changing attitudes to sex is key if infection rates among young people are to be reversed. Science may have moved on, but society is lagging behind, say experts. And AIDS is still a four-letter word that spells shame. Karen Allen, BBC News, South Africa.
All right, so guys, these are the type of things we're going to be covering in Unit 2. So I think on a whole, I personally like teaching Unit 2 biology because there are so many social discussions we can have. And it is a different type, I think it's a different perspective on biology now because it's now teaching you to think on a broader level and not just confine biology to just learning about cells and systems. You're now seeing the wide scope of biology by learning about biodiversity and ecology and learning about economic structures and social structures affecting disease. So it opens up your mind to see how biology can lead you into so many different occupations, all right? So let us see now how best to prepare for this. So starting from this week to the end of April, you really have 32 weeks of, well, 32 weeks. But in terms of teaching weeks, 6 of those weeks will usually be holidays. For December, Heroes Week and Easter break. So you really only have 26 teaching weeks. Now, usually we finish in 34 weeks. We complete the syllabus in 34 weeks. And so it means that because you'll have 1 or 2 weeks when you have a break, we do have to have 1 or 2 extra classes, usually in term 3, to ensure that we get in our 34 sessions to complete all the topics thoroughly. But it's usually a subject we can comfortably complete. In terms of your assessment, you are assessed with 2 papers. Yours paper 1 with 45 multiple choice questions, 15 from each module, and paper 2 with 3 compulsory structured essay questions, 1 from each module. Each question worth 30 marks, and that makes up 40% of your grade. And your labs consist the other 20%. So just like in Unit 1, in Unit 2, your paper 1 and paper 2 are weighted equally. Each module is weighted equally. So there is no benefit for doing exceptionally well in one module and not being competent in the other. The real thing is being competent in all. So this is just giving you some more detail about paper 1. All of the 45 questions are compulsory. And questions may be presented as diagrams, data, graphs, prose, or other stimulus material. And paper 2 has 3 compulsory questions, 1 from each module. It's 30 marks per question, so you get 90 marks for the paper. As for the labs, CAPE does not give a specific or a minimum number of labs. However, you must get at least 2 labs on each of these skills. So you must do 2 labs on observation, recording and reporting, manipulation and measurement, drawing, analysis, interpretation, planning, and designing. So in essence, you end up having to do, if you're doing 2 labs on each skill, then I would say that the minimum you must do is 10 labs then, right? Based on this logic. Okay. And then you have your final grade, and I think there's a challenge here. So that was the wrong slide. So remember now, your paper 1 would be 45 marks, paper 2, 90, and then that would end up being weighted. So the paper 1 will be weighted to 40%, paper 2 will be weighted to 40%, and your labs will be weighted to 20%. This is talking about if you did the 3rd paper, but we don't do the 3rd paper, we do labs. So guys, for Unit 2 Biology to do very well, you want to attend to the subject at least for 3 hours minimum. And you must do the questions, essay questions and multiple choice. As soon as you learn something, you want to see how the topic is applicable to questions. And what happens when you do the questions right away, it helps you to understand what parts you should zone in on. Because when you notice something is tested frequently by CXC, then it tells you that I need to study this especially carefully. You don't have that level of insight when you don't do questions. If you're just depending on just learning the topic and just studying everything, you won't know which areas are more applicable, which areas are high yield versus which areas are low yield. Only doing the past papers frequently give you that information. And by doing the questions and also submitting them for us to mark it and we give you back the grade, it is also giving you an opportunity to increase your exam writing skills or your examination skills, because you'll find that once we are using a CXC mark scheme to grade you, when you submit something which you thought was adequate to get the points and you are not getting the points, then it forces you to improve. It forces you to give more points. It forces you to draw diagrams and graphs if you're trying to score as many points. So it is essentially grooming you and training you in that process each week when we mark your papers and give you back your grade. And so by the end, we do the marathons and we do the questions another time, then by that time now you are way more sensitized to know exactly what CXC wants from you to get the marks, right? Because in biology, it's usually that you're not really struggling to write an answer, you see the lines and you usually think up something and you usually write something in the space. But what usually happens is that your answers are not high yield. They're not high yield. And that's what we are trying to avoid. For you to get the one, you have to have targeted specific answers, which are high yield answers. And only practice of past papers do that. So of course, your final grade will be, the profile will be for each module, module 1, 2, and 3. And you want to be competent in all 3 modules to score that grade 1. Make sure you have your personal copy of the syllabus. Make sure you're ticking off the objectives that were covered each week in your syllabus. And also remember, guys, that even though CAPE doesn't pressure you to do more than 10 labs, they do have suggested practical activities with most or several of the objectives. So even if you don't do a lab on that area, because CAPE has it in the syllabus that there is a suggested practical activity, means they can very well test you on that lab. They are testing as if you did it. So for example, when water ascends in plants, it is done by root pressure, capillarity, cohesion, adhesion, transpirational pull, and you are to include the role of the stomata. So this is what they want you to know about this objective. But then they say there is a suggested lab. You could look at wet mounds of celery that were placed in colored fluid, and you could use the coleus epidermal peels to visualize tomatoes. So this is what CAPE suggests you could do as a lab. But we're saying that even if you never did these labs in school, because they suggested it, it means that a question can come on it. Multiple choice question can come, or another question can come showing the celery mounts that they described, and you never did it. But they're expecting that once they put it in the syllabus and put a suggested practical activity, that you're actually going to research it. You're going to look it up on YouTube or something so that you have seen the lab. Make sure you read ahead before you do experiments. Unfortunately, sometimes because of time constraints, the teacher might give you a lab on a topic that you have not yet been taught. So that I know is not the ideal situation, but you can quickly go on YouTube and research it and visualize it and hear how it's discussed, so that when you do it yourself, you have a better insight, because the labs are not just there to stress you. They're not there to stress you out at all, guys. It's there to reinforce the topic that you're learning. So it really should be enhancing your learning. Make sure you watch YouTube videos and make sure you visually look at labs and get insight. For example, this one here about the dye moving in the celery, you may not do it in school, but you could have gone on YouTube and watched it and seen how it's done and seen how it looks under the microscope. So if you see this in a question, even though you never did it, you have idea about it because you saw that it was a suggested practical activity and you researched it. So please remember that you are expected to do that, especially at the CAPE level, which is why you must have your own copy of the syllabus. Because if you don't have a copy of the syllabus, then you're not going to know what suggested practical activities there were for which objective. You're going to just depend that the teacher is going to be researching it for you and showing you. And I don't think you want to leave up your grade like that. So I implore you to have your own copy of the syllabus and follow along with the teacher. Make sure that you're completing each module, you're completing each objective, you're researching the practical activities, you are doing the past papers, multiple choice, and essay as you go from topic to topic. Once you do that, guys, there's no way you're not going to score a grade 1. So those are my suggestions to you for Unit 2 Biology. Do you have any questions? Any questions? So at Extra Time, we actually do start our classes next Sunday for Unit 2 Biology. If you need any extra help, we do have classes for it. But thank you so much for attending. And if you had signed up for the Unit 2 Chemistry, that's going to start at 5:45. So in another 30 minutes, you can log back in for the chemistry if you had signed up for that. But thank you very much for attending, and I hope this has been useful for you.